To power sustained flight, a bird relies on an internal architecture engineered to store and mobilize extensive lipid reserves without suffering metabolic collapse. Mammals distribute the work of fat synthesis across multiple tissues. Avian metabolism centralizes fat production almost entirely within a single organ, creating a singular, high-capacity hub for fuel generation. We can map this specialized pipeline by tracking a single lipid molecule from the moment of ingestion, routing it through a specialized bottleneck in the liver, packing it into high-density storage, and finally driving it into the muscle cell for mitochondrial combustion. Tracing this exact pathway explains why commercial poultry succumb to fatal metabolic diseases under high carbohydrate loads, and it reveals the specific biochemical sequence that allows migratory species to cross oceans without stopping. The absorption sequence begins in the avian duodenum, where dietary fats are emulsified by bile salts and broken down by lipases. Once inside the mucosal enterocytes, these cleaved fatty acids are immediately re-esterified with glycerol, rebuilt into full triglyceride structures. To move these highly hydrophobic triglycerides through the water-based blood, the enterocyte packages them with a dense array of structural proteins to form a large transport particle called a portomicron. Mammals package dietary fats into chylomicrons, which enter a well-developed lymphatic system to bypass the liver initially. Birds lack this extensive lymphatic network. Instead, the avian portomicrons exit the enterocyte base and are dumped directly into the portal vein. This direct route means the liver receives the entire incoming load of dietary lipids first, rather than the mammalian model where fats are systematically dropped off to peripheral tissues before liver clearance. Because almost all dietary fat surges straight to the avian liver, this single organ acts as a concentrated physiological bottleneck for lipid processing. In mammals, liver and adipose tissue synthesize fat. Avian adipose cannot. The liver bears the entire metabolic load. Inside the hepatocyte, precursors become acetyl-CoA. Acetyl-CoA carboxylase converts this to melanyl-CoA, feeding the fatty acid synthase complex to extrude long fatty acid chains. These newly synthesized fatty acids are re-esterified into triglycerides, bundled with ApoB100 proteins, and packaged into very low-density lipoproteins, or VLDLs. When poultry are fed diets with extreme carbohydrate-to-protein ratios, this centralized system is forced into lipogenic overdrive. The excess fat accumulates in the liver faster than it can be exported, resulting in severe hepatic steatosis or fatty liver syndrome. Under normal conditions, the liver efficiently secretes these VLDLs into the bloodstream to transport the newly minted lipids out to the rest of the body. The avian liver functions as the exclusive production factory. Every other fat depot in the bird's body operates as a high-capacity storage warehouse. VLDL particles exit the liver, navigating systemic circulation toward highly vascularized fat pads. At the capillary's luminal surface, the enzyme lipoprotein lipase, or LPL, acts as a gatekeeper. Anchored to the wall, it hydrolyzes passing triglycerides housed in the VLDL core. This enzymatic cleavage releases non-esterified fatty acids, which readily cross the membrane into the adipocyte. Once inside, the fatty acids are reassembled into triglycerides one last time, aggregating into a massive lipid droplet that occupies over 80. A bird's fat store volume relies entirely on the efficiency of its hepatic factory and the regulatory pull of lipoprotein lipase at the destination. In the weeks leading up to migration, continuous lipid deposition triggers pronounced adipocyte hypertrophy. The fat depots swell, transforming the bird into a fuel-dense projectile. To burn this stored fuel, the bird must reverse the storage process. While mammals rely on catecholamines to trigger fat breakdown, birds utilize glucagon as the primary hormonal signal to initiate lipolysis. Glucagon activates a cascade of lipases, starting with adipose triglyceride lipase, which systematically cleave the stored triglycerides back down into individual fatty acids. These non-esterified fatty acids exit the adipocyte, diffuse back into the bloodstream, and immediately bind to circulating albumin proteins, which act as molecular transport shuttles, ferrying the fuel directly to the skeletal muscle. By bypassing the lymphatic system and centralizing synthesis in the liver, birds achieve a turnover rate far exceeding the mammalian baseline. While rodents may take over a week to cycle through half of their stored lipids, the highly efficient avian system enables a rapid shift from building fuel stores to exhausting them for sustained flight. The albumin carriers deliver their payload to the avian flight muscle, specifically the red, slow oxidative fibers packed with capillaries designed to run continuously for days. To be burned, a specialized carnitine shuttle pulls the long carbon chain across the inner mitochondrial membrane. Inside the matrix, it undergoes beta oxidation. It is systematically clipped 2 carbons at a time, generating molecules that feed the Krebs cycle. This sequential combustion yields roughly 39 kilojoules of energy per gram, more than twice the chemical energy extracted from burning an equivalent weight of carbohydrates or proteins. This specialized pipeline provides a consistent stream of energy-dense fuel, turning fat into a lightweight power source capable of sustaining a 3,000-mile migration.